Hello everyone, good afternoon all. It's Gautam and I'm taking botany here. And we have completed already one session in the last what we are going to discuss. What we have already discussed is sexual reproduction in flowering plants. Introduction part related to that I have a bit discussed something related to that. So we have started and we completed one page of there. And before that also we have seen there regarding the introduction of the unit and regarding introduction of the scientist there. See, when it comes to the NEET aspect, keep in your mind that everything is important. If you check in last year 2025 PYQ, you will get to know that even scientists also important there. So in that they have given, see, there are not much scientists are involved. See, if you take 11th and 12th bio books, there are 10 units are there. So in that, sorry, 9 units are there. In the beginning of each unit, there you will be seeing a introduction para of that complete unit, and along with that, you will be seeing an scientist introduction there. So that's not, you need to give importance to the each and everything there, including the introduction and even including the scientist introduction also. So in the last class, I discussed about scientists there, Panchanan Maheshwari, which is given in the beginning of your unit, reproduction unit there. So Panchanan Maheshwari, who worked, who contributed in Indian plant embryology, and his contributions and his achievements we have discussed in the last class. And along with that, we have seen the introduction of reproduction unit. So unit of reproduction, it includes three chapters where sexual reproduction in flowering plants, human reproduction and reproductive health. So in that first chapter we are going to discuss in plant science is sexual reproduction in flowering plant. So this chapter has got this much of importance where you can see the past year analysis of this chapter. So that there will be minimum 2-3 questions will be there, the maximum like 4-5, it extends to 6 also. So where it carries a lot of weightage. This is the past 10-year analysis of questions you can see here. So from this chapter, these many questions, these many number of questions has been asked, and this is a topic wise analysis. This topic wise analysis, you will get to understand after after the completion of this whole chapter, we can come back and see that which concept has more weightage. And before studying also, we need to understand that which has more importance, which has less importance, so that we can emphasize or we can give a proper focus on that particular concept. So we need to analyze everything on topic wise based also. So here in the last class I discussed about the formation of flower. So how the flower is formed in plants. See, uh, flower is flower formation in plants. So how it will take place and what are the things it will be taking place within the plant, everything we need to understand. Just to think, see, in a plant, uh, in the last class also I said that if a plant is very young and green, no flowers are there, but when it comes to the spring season or the particular season where they will bloom flower, where they will start to flowering, so that before starting flowering, they will be there will be some decisions will be made within the plant, so that decisions are not discussed with the family members, so that is an internal decision. So if you are studying a need there, that is the internal motivated guy who is studying for that particular exam. It means internal motivation should be there. So I need to stand on that position, and by keeping in mind that, so we will be evolving and modifying within ourselves. So it is the change of individual, it is the change of within ourselves. It is nothing but it is not the change of someone else. So I need to take decision from someone others and someone else to consider my own future. See, if I want to flower myself, if I want to bloom flower, or if I want to flowered and look colorful and beautiful, it is one who you to decide. Means I need to decide if I want to look brightly colorful and to appear very nice, to have a great future. I need to decide myself. So like that, coming to this, in plants also they will be deciding within themselves with a physiological decision, by changing the hormonal conditions, everything, structures it is going to change. So now if you see here in formation of flower, so the flower formation it will be decided before only. So if a plant is there, so this plant it will decide the flower formation before itself to before it starts to flowering. Okay. So it will be the decision will be made internally. So how the decision will be made? Before this, what it has? It is having a shoot apical meristem. So shoot apical meristem is there, which we have already discussed on this. I am just revising the things, so don't get confused there. So some of you have not attended the last session. Shoot apical meristem is there. So meristematic cells means what? Which continuously divides. So here which is present on the shoot region, which is present on the apex in the end of the shoot, that is shoot apical meristem, which shows a continuous dividing. So now this shoot apical meristem it modified into floral meristem. So it modified into floral meristem there. So that is nothing but it is modifying into floral meristem. Means still it has dividing capacity. See, meristematic cells itself has a tagging which are continuously undergoing division. So keep in your mind that it is the floral meristem. Floral meristem in the sense, so the flower is going to form from these meristematic cells. Here I am calling this as shoot apical meristem because these meristematic cells which are showing division is going to form shoot. This is floral meristem, which means the meristematic cells which shows a division will are going to form flowers there. So this floral meristem will undergo structural and hormonal changes. Structural and hormonal changes it will be taking place. So structural and hormonal changes it will be taking place, and it shows differentiation there. So differentiation means what? So you know already about these things because in 11th standard plant growth and development topic you will be learned about differentiation. Differentiation in the sense the cells which are meristematic means which are showing the capability of division will lose the capacity of the division to perform a specific function there. So this floral meristematic is the cells which are showing division will lose the capacity to divide, and it will be showing some particular structure there, that I am calling this as floral primordium. So here I am calling this as floral primordium. Primordium in the sense initial structure of a flower. So if a flower is going to form very colorful like this big one, before that it will be like this. It is the floral primordium which is not yet formed, which is still it is in the bud stage. So this is the floral primordium. This floral primordium will shows it will bloom and it will becomes flower later. So this is how the formation of flower will be taking place. So floral primordium is formed after differentiation. So here it is having meristematic activity. So now it has lost the meristematic activity and it has got some functions there, that I am calling it as floral primordium. So floral primordium is nothing but the flower initial structure. So it is the initial structure of this one. So this flower initial structures will lead to form inflorescence, or if it is the flowers are forming in bouquet, means they are arranging. Inflorescence means what? This also you learned in morphology of flowering plants. So the arrangement of flowers on the axis, I am calling it as inflorescence. So if a plant is showing many flowers in an axis, I am calling it as inflorescence. If a plant is showing only one flower or on the branch or on that particular branch, so it will be showing it in the form of bud there. So either it will be showing in the form of inflorescence or in the form of bud. So this it will lead to formation of flower there. So this is what we have completed in the last session, and the cultivation of that one. So the same thing here. So cultivation of flower is nothing but it is a cultivation of flower is nothing but floriculture. So that we have already seen in the last class. So now next, so the structure of flower is also discussed. So there we have come across four whorls. The structure of flower, that is we have already discussed there. So where it will be having four whorls, two are reproductive whorls which are directly involved in the reproduction, and two whorls are protective whorls which are covering the reproductive part. So that is like calyx and corolla, they are covering the androecium and gynoecium. So they are covering and protection, giving protection to that. And we have discussed the different events of sexual reproduction also. So events of sexual reproduction in the sense pre-fertilization, fertilization, and post-fertilization. So that an outline of that one we have discussed in the last session. If you didn't watch, you can go back and watch that session also. So continuation of that, we will be seeing a pre-fertilization structure and events. See, in the word itself is there pre-fertilization. Pre means before the first, before the fertilization, that is pre-fertilization. After the fertilization, it is post-fertilization. So fertilization is in between that it will be there. So before fertilizations, what are the structures we are going to see, and what are the events we are going to see, that we are going to learn in pre-fertilization and structure and events there. So here, coming to this, in the last section itself or last session itself, I said about this, that is pre-fertilization is nothing but where gamete formation will be seeing. We will be seeing gametes and how the gametes will be formed. So coming to this, first one is we'll move from the male reproductive structure. So that is male reproductive structure or androecium. So I will be writing with A, the A term there. A indicates androecium there. So A is androecium. So here male reproductive structure, what and all it is involved. So let me draw that one. So and so it is stamen there. Androecium is nothing but it is the stamen. So it will be connected with this one, a long slender filament like structure. It is long slender like filament structure. We call this as a filament. So it is a filament, and this one is anther. So together we call this as stamen. So here this one is anther, and from here to here it is there, no? From here to here. So from here to here we call this as this this part from here to here, we call this as filament. And the thing we need to note here is, here anther is attached to this filament with the connective tissue. Here the point of attachment is there, no? This point, it has connective tissue where it is connecting to this anther there. Okay. So now this filament, this end is there, no? This end I am calling it as proximal end. Okay. So this end is proximal end. So this, it is a proximal end. Proximal end in the sense what? You might be hearing that term, even in the 11th also it has come, this word has come in many, it came in many chapters there. So proximal in the sense, see, this is my, this hand, it is radius and ulna bones will be there. So this is the point of connection, so which is from where it is originating, and it is near, no, this point, this joint. So it is a proximal end, I can say. So like that, so this proximal end of this filament, it is connected to, so either it will be connected to thalamus there. So the proximal end of this, it is connected to thalamus. So proximal end, either it will be connected to thalamus, or in some plants, like if you see the flower of tomato, brinjal, or if you see the flower of brinjal also, you can get to know that where the stamens, it will be attached to the petals there. So in some it is attached to the petals, and in some, like if you see the if you observe the lily flowers, so in flowers, lily like flowers, this end of this proximal end of the stamen is there, no, it is attached to perianth there, or tepal we call it as. So if it is attached to the petals, then we call this as epipetalous condition. So you know about this one, it is epipetalous. It is epipetalous condition. So perianth, if it is attached, it is epiphyllous, we call this as. It is epiphyllous. So thalamus, they don't have any specific term there, because in general the stamen's proximal end, it will be attached to the thalamus. So these are the some specifications where it will be attached there. And the length of the stamen, the length of the filament, it varies in different species also. That also you have learned in morphology of flowering plant. So I might be saying you learned already, no? So let me connect to that. You just check, you will get to know in biology class 11, so morphology of flowering plants. See, inflorescence, we have just now I said, no, inflorescence word from here. So next question, where it is now. See here, in androecium, if the stamens of flower may be united, okay, leave that stuff. When stamens are attached to the petals, I just now said, if the stamens are attached to petals, it is epipetalous condition. So here it is epipetalous, here it is epipetalous there. And if it is attached to the, in case of lily, it is attached to this perianth, it is epiphyllous condition. So it is epipetalous condition. So it is from 11th chapter, you need to remember this, all these things. So even I said length of the filaments, it varies within the flower also, within a flower like salvia and mustard. The length of the filament in androecium, I am not discussing anything here. I am just revising the morphology of flowering plants content linking to this sexual reproduction of flowering plants. So this length of the filament, it varies even different plants. So even why I am revising, Even in when you are reading the NCERT of 12th also, you will get to see. So this length of the filament, so length of the filament, it differs in different plants. Varies in the same plant or within the plant. So example for that we have seen, no, just now salvia and mustard. So you can write here salvia and mustard. And this one epipetalous conditions like brinjal, I said, no, that is brinjal. And epiphyllous conditions lily, where the perianth it will be attached, and thalamus in general it will be there. So now coming to this answer now. So answer, how it looks like here. So if you see the answer shape and size there, so the shape, it is having two lobes there. It is having two lobes. So the answer, it is, so here, so it is answer. Answer is bilobed condition. Bilobed means two lobes are there. This is one lobe, this is another lobe. It is, this answer is in bilobed condition. Okay. Bilobed condition. Bilobed in the sense two lobes are there. And one more thing you need to note here is, in each lobe there will be two theca will be there. Two theca in the sense THECA, theca in the sense nothing but bag or sac. Sac in the sense bag which is containing within that the pollen grains it will be containing. So the some bags will be there which encloses pollen grains within that. So this is bilobe and dithecous. So it is bilobed and dithecous. Dithecous in the sense it is having what? Two theca. Two theca in each lobe. So in each lobe, two thecas in the sense how many theca it will be? You need to answer. So how many theca it will be there in the this time and here? How many theca will be there in this one answer? There will be four thecas will be there. So there are two thecas in each lobe. So two thecas in each lobe. There are two lobes are there. Then it will be four thecas will be there within the one answer there. So it will be bilobed condition and dithecous. And one more thing which is given in your book. So if I am cutting the, if I am cutting this answer, if I am cutting this answer like this, so it will be having a transverse section. So the transverse section, it will be showing a polygonal structure there. It will be showing a polygonal structure where four angles are formed. That I will show in the next slide. But I am writing the information here itself. So answer is bilobed dithecous and it is a polygonal shape. I will show what is polygonal and how it will be. And it is also tetrasporangiate, also we call this. I will say what it is. So don't worry about these two words. I will discuss in next slide. So it is, answer is bilobed, dithecous, polygonal and tetrasporangiate in shape, and it will be, if you cut that, it will be, you will be seeing all these things. So now next we will be discussing that structure of this answer when you cut that one. So answer is there, no? So when the answer is young, when answer is young, see I am using the theca word, no, theca is nothing but bag, it is nothing but sac. So here this theca is nothing but bag or sac. So it is nothing but bag or sac, isn't it? So if two thecas I am using, so at the same time I will be calling it as sporangium also. I am calling the same bag as the same bag as sporangium. So it is depends on the content which is filled within the bag. So if a bag is there, if I am filling something vegetables in that, it will become vegetable bag. If I am adding that fruits into that, it will become fruit bag, like that. No, the bag is the same, but the name it is changing based on this here. See, if theta I am calling means it is just enclosed with the some tissues there, some normal tissue, and sac also you can call it as. And if it is having sporogenous tissue, I am calling it as sporangium there. If I am, if this is, if the same bag, if it is containing pollen grains, then it will be a pollen sac. Okay, so understood. So this is based, the names are based on the presence of the things which are present within the bag. So I am calling the same bag with a three different names there. That itself. Okay. So now next I will be seeing the structure of this one. So this diagram is of your NCERT diagram. You can see this is two lobes are present here. So when you cut these two lobes, you can see this structure. So here you can see this structure. So when you cut these two lobes, the transverse section, it will be having this kind of shape. So the same one I am drawing here, the answer of this one. So if the same diagram, if I want to take this, the same. So there is some technical error. Wait for it. Wait for it. Okay, you can see here. This is the structure. So when you enlarge that one, the same here, the transverse section of microsporangium I am drawing here. So here, okay, the same structure I am displaying here. So it is a transverse section of microsporangium, we call it as. Microsporangium in the sense, see, the anther is there. So it is, when I am cutting anther, I can see the two lobes. So two lobes in the sense, see here. So it is, these are the two lobes. So it is two lobes there. So these are the two lobes you can see here. And within that you will be having sacs in the sense. So as we have already discussed, sacs are nothing but small bags which are present within that. So these small bags which are present within that. So these are the small bags which are present there. So these are the small bags which are present within that. See, in each lobe there are two thecas are there. So the two thecas in each lobe, I can say it as four pharyngium, four bags. Means see, one theca is nothing but one sporangium. This is another sporangium. This is another sporangium. This is another sporangium. So if you check here, so there it is organized in the form of, see, you can see here, it is organized in the form of four angles are there. So it is polygonal. So polygonal structure it is. So it is having polygonal structure, and four sporangium are there. That is a tetrasporangiate. So in the last slide I discussed about this. So I wrote that it is having polygonal shape. So it is having polygonal shape and tetrasporangiate. Tetrasporangiate means what? Four. Four means tetra. Tetra means four. So sporangiate, sporangium. Four sporangium are there. Okay. So that's four sporangium are there here. So now this sporangium in when it is in young stage, when this in young stage, it will be having a normal tissue, like upper all the parts of it is having not the same normal tissue it will be. So now this it is the main important organ now that where it is containing the proper pollen grains. Pollen grains in the sense we also call this as microspores. Micro means small. So how the pollen grains will be? It is comparatively it will be smaller if you see the ovules or ovary. So that's why we call this as a microsporangium structure. So it is a tetrasporangiate, and this we can call this as a um, TS of anther also. TS of TS in the sense a transverse section. So when I am cutting like this, it is a transverse section of anther, or it is a transverse section of microsporangium also you can call this. And what about remaining part? What will be present in the sense? See, at the center you will be finding some structure here, that is the connective tissue where it will be connected, and means where the filament will be connected to that. And these are the four sporangium, and each sporangium around this, so it will be surrounded by some cells there. So some cells are surrounded upon this here. So these are surrounded by some cells. So these are some surrounded by some cells there. So these are some cells are surrounded. Around to that another layer is found. So another layer you will be found, finding around another layer around this one. So there is another layer is found. So another several layers are found, that is we call this as middle layer. Several middle layers also found, and upon that we will be seeing another layer that we call it as endothecium. So now what I am discussing is TS of anther and the layers which are present around that. A layer of cells which are present around that. That is endothecium. And finally there you will be seeing this layer. So now I will label these cells where you can see within that what you are seeing, these cells are there, no? These are tapetal cells, we call this as the innermost layer or the innermost cells of this sporangium or TS of anther. These are tapetal cells. So these are tapetal cells, and this layer we call this as tapetum. And some cells has 2 nucleus. The cells of this, it is the one of the important a topic in this 12th class, not only with aspect to the board, but majorly it is very important for NEET exam. Even last year also they asked on this, on the tapetal cells. See, what I am discussing here is, we are discussing TS of anther. So when you cut the anther in a transverse section, you will get this shape. And this TS of anther or TS of microsporangium also you can call. So these are the layers of which we see in each theca or pollen sac or microsporangium there. So we will be seeing these many layers. So this, which is present inside of this, no, this one. So this here, which will be present, so it carries all the cells. So initially it will be having only sporogenous cells. Sporogenous cells it will be having. Sporogenous cells in the sense, sporogenous means what? So it is having, the cells will be having diploid content. So diploid content of chromosomes means if you take in my body this cell of this one, it is having 46 chromosomes, means 23 pairs it is. So here this, it is the sporogenous cells, not gamete cells, sporogenous cells, and the cells together I am calling this as sporogenous tissue. This is sporogenous tissue. This is sporogenous tissue. And surrounding to cell, sporogenous tissue, surrounding to the sporogenous tissue, what you are seeing, the yellow color layer is there, no? That is the innermost layer of this anther. That the innermost layer means exactly surrounding to this sporogenous tissue is there, no? That is the tapetal cells or tapetal layer, we call this as. This one is tapetal layer, or the cells we call it as the tapetum cells. And the special thing about this tapetum cells is, see, these sporogenous cells, they are going to form a pollen grains there. See, in future means when this TS of anther, it is a young anther now, so that's why I am drawing sporogenous tissue. So when it get matured, so it is going to form pollen grains there. So then I will be calling this microsporangium as pollen sac. So if it contains pollen grains, it is pollen sac. If it is containing microspores, it is microsporangium. If it is containing sporogenous tissue, it is sporangium or microsporangium. Okay, so you can call this as theca also. So surrounding to that, the yellow color layers, they are made up of tapetum cells, they are tapetal cells. And the special thing about these tapetum cells are, they are dense in protoplasm, like abundant of food materials will be there, so that it helps these pollen grains to develop. It nourishes the pollen grains, it helps in developing of these pollen grains. When it supplies the food like this towards the sporogenous tissue, so when it supplies the food here, so it is the developing, these are the developing pollen grains. So these are the developing pollen grains. The food will be, to develop the pollen grains, the food will be supplied from these tapetal cells. The innermost cells will be nourishing the pollen grains there. So the innermost layer, they are nourishing these cells to get matured into a pollen grains there. So the function of tapetum cells is very important in this case. The function of tapetum cells, it helps the nourishment, giving nourishment to that. And next to that tapetum cells, what you are seeing, the two blue color lines, the two not only two, it will be two to several layers, it will be, that we call it as the middle layers. And the outermost layer is there, no, the green color one, this outermost layer is next to that, this layer is there, no, this layer is epidermis, we call it as. In plant anatomy you have come across, epidermis means the outermost layer is epidermis, and next to that, the green color layer which you are seeing here, this one, the green color layer is endothecium. Related to the functions, we will discuss that later. First, let me write this. Uh, what are the different layers we come across? So there are four layers are there. One is the tapetal layer, the innermost layer, and next year it is the next two layers are middle layers. These are middle layers, and next to that it is endothecium, and next to that there will be another one more layer will be there, that is epidermis. So these are the several layers which we will be coming across. So that is, one is epidermis, second is endothecium, next to that the third one is middle layers, and next to the innermost layer is tapetum cells, and inner to that the sporogenous tissue will be there. So it applies to the all thecas. Okay, this is just four wall layers of I explained of only one sac here or one microsporangium there. So it applies to all four there. So next to that we'll discuss the functions of that layers of that one. So it is a structure of microsporangium, and the functions when it comes to this function here. So here you can see this. And the cells which are present within that, all the cells are sporogenous tissue. Okay. So now I will label the parts, along with that we'll discuss the function also. The outermost layer is epidermis. So it is epidermis. Outermost layer we call this as epidermis. And next to that what you are seeing, that is endothecium. So this layer is endothecium. So this layer is endothecium there. So next to that is endothecium, and after that you will be seeing a several layers there. So that we call this as middle layers. So next to that you will be seeing middle layers there. And next to that you can see that there is a difference between this group of cells and the surrounding cells. There is in between that there is somewhat dense structures is seen this part. So this part you can see, in this part there, in this part there is somewhat something is there darkly stained, which is a darkly appearance you can see there. So it is somewhat having some granules within that, and which makes appear somewhat like clumsy. So that is that layer is nothing but it is a tapetum layer there. So that layer is here. This layer, it is a tapetum layer. So this layer, it is tapetum layer. So it is tapetum. So now coming to the functions of this one. So epidermis, even though their first three, epidermis, endothecium, and the middle layers, they work together as a protective unit. So their individual function somewhat differs. So here epidermis is the outermost layer. So it is here, epidermis is outermost layer or the first layer, which helps in protection. And next layer is endothecium. Endothecium is second layer, and it is hygroscopic in nature. Hygroscopic in nature. Hygroscopic in the sense, see, when the anther is young, It is somewhat green and somewhat nice, like wet it will be, wet it will be. So when it is about to dry, means when it is about to dry, so the for the pollen grains dispersion, it has to lose the water moisture. So means that losing of water moisture is nothing but hygroscopic there. So drying out or losing the moisture, losing moisture and forming dry there, drying out. So this, it is nothing but hygroscopic, and hygroscopic, these cells are hygroscopic and it helps in losing moisture, and it will get dry. So when it get dry, the anther will burst. When it bursts, the pollen grains which are present, that it is going to shed. The pollen grains going to release, and it helps endothecium. It works as a protective unit also. So it helps in protection also, it helps in protection also, and it also helps in releasing of pollen grains. Or how it will release, how it will be releasing, in the sense it will helps in dehiscence of anther. So dehiscence of anther, in the sense, so dehiscence is nothing but drying out and bursting and releasing the pollen grains. That is nothing but dehiscence of anther. So there are several middle layers also there which are helping in protection. So next comes the tapetum here. So here tapetum has a specific function. So this tapetum has a specific function there. So where, so it is, this is tapetum. So tapetum. So when we discuss about the tapetum cells, so how it looks when you observe under microscope, tapetum cells, they will be having dense protoplasm or dense cytoplasm. So they will be having dense protoplasm there, and it is binucleate structure. So it is having binucleate structure. Binucleate in the sense the cells will be having two nucleus. So two nucleus is found. So how the two nucleus is found? So usually in a cell one nucleus will be there, but in tapetal cells two nucleus are found. So you need to connect to the topic of cell cycle and cell division there. In cell cycle and cell division, you will be learning there when, see, when cell is undergoing cell division, so that it forms a two nucleus. So after that the wall formation will be taking place. So when the wall formation is taking place, so then the two cells are formed, two newly cells are formed. So usually cell division has two parts there, or two stages we call it as, or two phases. One is karyokinesis. Karyokinesis means nuclear division, and the next is cytokinesis. Cytokinesis means complete wall formation, making them two cells. That is cytokinesis. So here in case of tapetum cells, there is no wall formation is taken place. Due to that, it got two nucleus within the same cell there. So it is binucleate. Binucleus means two nucleus is seen in tapetum cells there, and dense protoplasm is seen. So it is appearance. It is about their appearance. But the coming to the function here, the function, it got a very good function there, where it will be nourishing to the developing pollen grains there. So it gives nourishment. It gives nourishment to developing pollen grains. So it is like a giving nourishment to the gametes to develop, to continue the development there. So it will be having some food storing, abundance of food storage will be there because already dense protoplasm, in the sense it has some very good amount of food. So it will give that food, that in the form of nourishment to these developing pollen grains, because it is within that the sporogenous tissue is there, no? So that it will give that. Okay. And next to that, tapetum cells, inner to that, what we are seeing. So these all are sporogenous cells. So all these are sporogenous cells or sporogenous tissue. So it is a sporogenous tissue. Sporogenous tissue in the sense it is sporogenous cells. So these sporogenous cells, after taking nourishment and after developing, these are going to form pollen grains. So these are going to form pollen grains. So if it is containing sporogenous cells, or at the time I am calling this structure as microsporangium, because it is containing what? Microspores. If it contains microspores within that, I am calling it as microsporangium. So if it contains pollen grains, then I am calling this as pollen sac. So at that time I am calling this as, within the pollen grains will be there within the pollen sacs. The sporogenous cells or the microspores will be there within microsporangium structure. So this is about the structure of microsporangium, or TS of anther we call this as. So related to this, we can discuss some questions there. So remaining things we can discuss that later. So the coming to the NCERT lines. So these are the NCERT lines given in your book. So these are the NCERT lines which are given in your book there. So structure of microsporangium, it is a microsporangium is nothing but the theca which is carrying microspores within that. That in a transverse section of typical microsporangium appears near circular in outline. See here, it was appearing in a circular in outline there. See, it is circular in outline. So they are talking about there. So the NCERT, it is explaining about the circular in outline. It is structure of microsporangium. And it is surrounded by 4 wall layers. So it is generally surrounded by 4 wall layers we have seen already, that is epidermis, endothecium and middle layers and tapetum. And the outer 3 wall layers perform the function of protection and help in digestions of anther to release the pollen grain. Means it helps in digestions, means digestion, here digestions in the sense drying out, losing the moisture and helping them to release the pollen grain. And the innermost wall layer is tapetum here. The innermost wall layer is tapetum. And coming to this, nourishes the developing pollen grain. And the cells of tapetum possess dense cytoplasm or protoplasm and have more than one nucleus. So that is usually it will become binucleate. So when it will become binucleate, you can think there. Okay, let's go back to the NCERT. You will get to understand. I will show in cell cycle and cell division there. So that is nothing but cytokinesis will not take place. Okay, so coming to this, here in cytokinesis, here you can see there. In some organisms, karyokinesis is not followed by cytokinesis. Means karyokinesis means what? Nuclear division. Nuclear division. Here cytokinesis means cytoplasmic division and making 2 cells separate completely. So karyokinesis is not followed by cytokinesis. So it's not followed by the cytokinesis. As a result of that, it becomes a multinucleate condition. The same thing we are going to see here in case of tapetum cells. See, can you think of how tapetals could become binucleate? So we already thought and we linked to the 11th concept there, where karyokinesis is not followed by cytokinesis. So that's why it is going to form binucleate stage. And within that you will find a homogeneous cells of sporogenous tissue we will be seeing. So next of this we will be discussing in the next class how the spores, means how the pollen grains are formed. See, when you go and observe to the flower, you can able to see the pollen grains. In the next class, how that pollen grains are going to formed within the plant cell, we will be knowing that. So we'll discuss that in the next class. So let us see, is there any questions they asked in previous year? Okay, yes, this is what they asked in previous year. We'll discuss this one question and we'll wind up the session. Assertion: cells of the tapetum possess a dense cytoplasm and generally have more than one nucleus. That is true, no? Cells of tapetum possess a dense cytoplasm and generally have more than one nucleus. So it is having more than one nucleus. That is the correct statement. Why? So why it is having more cytoplasm and why it is having two nucleus? So you should get that reason in your mind before reading the reasoning statement there. Okay, now I got that why it is like that. Now I will read the reasoning now. Presence of more than one nucleus in the tapetum increases the efficiency of nourishing and developing microspore mother cell. It increases its efficiency if it is having more than one nucleus and more cytoplasm, more food, everything and all. So I can supply nutrition to the developing sporogenous tissue there. Okay, so cells of tapetum possess dense cytoplasm and generally have more than one nucleus. That is correct statement. So presence of more than one nucleus in the tapetum increases the efficiency of nourishing the developing microspore mother cell. That is also correct statement, and it explains the A. So both A and R are true, and it explains A. So it is about that. It is about for today's session. So till next session we'll be seeing. Okay, thank you. We'll see you in the next session. So hope you enjoyed the class. Thank you.